Bacterial growth curve. When a bacterium is inoculated into suitable liquid medium and incubated at suitable temperature and pH, the culture passes through different phases of growth. When bacterial count of such culture determined at different interval and plotted in relation to time, a growth curve will be obtained. That we will call it as bacterial growth curve. So in bacterial growth curve, first we will inoculate the bacterium into a suitable liquid media and we will wait for incubation. And during incubation, the bacterial count are made at interval after inoculation and we will plot a graph in relation to time and number of bacteria and we will get this bacterial growth curve. So this is the bacterial growth curve graphical representation and this is x-axis. X-axis is for hours or time and this y-axis for the viable count. Here we will can see the viable count and time. And bacterial growth curve is having mainly four phases. First one is the lag phase, log phase, stationary phase, and decline phase. In this lag phase, this is the first phase. Here you can see the number of viable count of the bacterium will become stable because just we have inoculated the bacterium to this culture media, that is liquid media. So what will happen? The bacteria have to adapt this particular environment. And during this lag phase, what will happen? This bacteria will produce or it will start synthesizing enzymes or other metabolics for the multiplication of bacteria. And just after this lag phase, you can see this is the log phase or exponential phase. During this phase, the bacterial growth will be exponentially will increase. And during this time, the number of bacterial multiplication will be maximum. So you can see this number of bacterial viable count will be increase exponentially. Just after to the exponential phase, this is the stationary phase. In this stationary phase, you can see the viable count will be stable because the number of newly dividing bacteria will be equal to the death of that bacteria. That means in here you can see the viable count will be stable. There is no increasing in the viable count because during the stationary phase, what will happen? There will have the nutrient depletion because culture media will have the specific amount of nutrient or limited amount of nutrient will be in the culture media and bacteria will utilize this nutrient. So they will have the less or depletion of the nutrient. And also the death of the bacteria will be equal to that of the newly formed bacteria. So in the stationary phase, the number of viable count will be stable. That means the death of the bacteria will be equal to the newly formed bacteria. And after to the stationary phase, this is the decline phase. Here you can see the number of viable count will become declining and the number of or the rate of newly formed bacteria will become less and the death of the bacteria will become more. So the viable count is declining in this decline phase, or this is also called as death phase. And why it is declining means because already you will have the nutrients. The culture medial nutrient will be already utilized by the bacteria. When the bacteria rate will become more, more nutrient is required. So already this bacterial we are giving a limited number or limited amount of nutrient. So that nutrient depletion will be occur. So bacteria will not get proper nutrient for their multiplication. And also during this phase, there will have the bacterial accumulation or toxins will be more in this culture medium. And due to the toxins and also due to the autolysis of the bacteria, the death rate will be more than that of the newly formed bacteria. So in decline phase, the viable count will become less. or declined or reduced. Bacterial growth curve of total count. In bacterial growth curve of total count, here we will count the total number of bacteria. Total number of bacteria means the number of live and dead bacteria we will count here. In viable count, we will count only the viable number of bacteria. That means only we will count only the live bacteria. Here in total count, we will count both live and dead bacteria. So this is the uh, lag phase. Lag phase will be same as that of the viable count. That means the number of bacterial cell will not increase. It will be stable here. And the second phase is the log phase. In the log phase, the bacterial total number of bacteria will become increase exponentially. And after this log phase, here you can see in this stationary phase and after this stationary phase, the graph will be continuously steady without any decline in the total number of bacteria. While in the case of viable count, the stationary phase will be stable, and due to the death of the bacteria, the viable count, the graph will decline. Here in total count, the graph will not decline here because we are counting both the death and also the live number of bacteria. This is about the bacterial growth curve of total count.
Subscribe to Simply Micromed. If you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms, hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.